Good afternoon. My name is Corey, and I'd like to welcome you to the 13th year of the CITRIS Research Exchange Seminar Series and 21 years of the CITRIS organization. We have hosted a remarkable lineup of technology innovators in person and are glad to see you all joining virtually today for the Fall 2021 series. Today, we are joined by Carolyn Patton. Dr. Patten is a neuroscientist and physical therapist who specializes in the assessment and treatment of motor dysfunction associated with aging and adult neuropathologies such as stroke. She directs the UC Davis Biomechanics, Rehabilitation, and Integrative Neuroscience Lab and co-directs the UC Davis Center for Neuroengineering and Medicine. Dr. Patton's research focuses on understanding the neural basis for human movement, investigating human motor control, and adaptation from a perspective of neuromechanics. Using concurrent behavioral and neurophysiological methods, her laboratory has developed techniques sensitive to motor impairment. Current work to be discussed in this seminar uses Gait as an assay of brain health to develop a predictive biomarker of subclinical pathology. Dr. Patton's research is supported by the NIH, NSF, Department of Veterans Affairs, and Healthy Aging in the Digital World, a UC Davis big idea. It is my great honor to welcome Carolyn Patton.
Thank you, Corey, for the very kind introduction and remarks, and also to those of you at CITRIS for the invitation to talk today. So let me just get my slides active here. All right, thank you. So my objectives today are to give a bit of an introduction, talk about Gait as a vital sign of human health, talk about work that's been done in our lab to develop an assessment, an efficient assessment of Gait that we call now the ABLE, the Assessment of Bilateral Locomotor Efficacy, discuss our work looking at mobile gait analysis, and also put out our invitation for collaboration with our lab. So first, in the area of introductions, I continue to feel that I'm a newcomer at UC Davis. I moved my lab here in 2018, and in the understandable flurry of moving an established research program across the country and bringing people with us, we had our heads down. And just as we were surfacing and having the chance to meet people, there was this worldwide pandemic. And we're still finding our way, navigating our way through that. But in the meantime, I have been able to assemble a very talented team. And one of the important aspects of my research program is that it is very deliberately multidisciplinary. And so as you look through our roster here of graduates, undergraduates, and lab members, you will find that we have engineers of all disciplines, neuroscientists, psychologists, medical students, and the like. And this has been really the mix that I have very deliberately intended, very intentionally made over the years in order to keep the conversation going in the direction that I think is productive. I am faculty at the medical school, but for reasons of space, my laboratory is located in a historic building on the UC Davis campus. There has been some restoration. These are pictures of our lab just as we were getting it up and going. Key element is we are definitely a lab that uses 3D motion capture. We center around an instrumented split belt treadmill. If need be, we can incline it to do interesting experiments. And we also have a number of other tools that we can use to measure human motion, human force production, neuromechanics. This is a quick video. This is an ongoing project that we have right now where we're doing real-time biofeedback. We're modeling joint kinetics in real time and using it as a biofeedback biofeedback signal to induce learning and adaptation. And one of the other key components of our lab is the motion is important, but we also study the nervous system. And TMS, transcranial magnetic stimulation, is a tool that we use a great deal and incorporate it into our experiments as a way to probe the nervous system. Okay, but today what we wanted to talk, I want to talk with you about gait and about human walking. This is, for those of us in the business, a very classic figure of the gait cycle, going from left to right, from initial contact, when the foot first hits the ground, all the way through the gait cycle to the next time that that same foot hits the ground. And there are a number of things that are happening in this period of time. Well, why are we interested in gait? It's a key function of human behavior. It's an important developmental milestone. It's a way for us to transport ourselves. It provides autonomy. As a neuroscience problem, it is really an elegant integration of the musculoskeletal and nervous systems. And so that gives us an opportunity to look at it, consider gait as a marker of brain health. We can take metrics of coordination, metrics of stability, metrics of transitions. There are many forms of transitions, from one leg to another, from a stance to a swing phase, also from different gait patterns, from a walk to a run, or from a walk to a very slow walk. There is increasingly in the literature reference to gait as the sixth vital sign. And when this reference is made, it is usually in reference to gait speed, measuring gait speed, or the speed at which we walk. So obvious and observable and measurable parameter, but it ends up being a very limited parameter. So the point of this illustration is to think about all of the different ways that we can measure gait, measure human walking, from observation. We can measure activity. This is something that's done a lot. It's become increasingly popular to put wearable devices on people and measure steps, step counts. And from that, it is possible to get an estimate of gait velocity. Step watch is a wearable that was a precursor of the more commercial grade devices that we have these days. It looks a little bit like a house arrest device that is worn, but still provides fairly accurate data. And these current items are certainly a lot more appealing to the consumer to use. But when we get into really wanting to understand gait, understanding the coordination pattern, we need to take on parameters like spatiotemporal parameters, step, stride, cadence, stance, swing. Kinematics are the joint angles, and these can tell us about coordination either within or between limbs. Also looking at the mechanics, just as a generic term, but if we are able to measure grand reaction forces, we're then able to calculate joint kinetics, forces produced at a given joint. And with this level of information, we now have a lot more insight as to what is behind a walking speed. For example, if somebody has a slower walking speed, is that because they are impaired in their force production mechanisms? Or is it because of pain, or any of a myriad of reasons? An additional form of measurement is to measure their neural control, which can be done with electromyography, as I mentioned earlier, transcranial magnetic stimulation, EEG. There are people who are doing this in very elegant ways now. And if we go back here, I'm circling back on this concept of gait as a vital sign, gait speed as a vital sign. It can be obtained with a variety of methods, from stepwatches or data loggers all the way up to the full instrumented gait lab. And we now need to look at what's involved in measuring gait. If we're looking at observation, the practice observer can make good estimates and good descriptions of gait independent of location. With a practiced eye, a a trained observer can make very insightful decisions and assessments of gait. They can't measure speed concretely. But as we go down this continuum, we require more and more instrumentation, more and more fixed locations. And there are challenges with that, both the time and effort involved in studying gait, and studying gait and getting parameters. And the flip side of that is getting people to come and participate, and the accessibility of the data. But there are huge advantages if we look at biomechanical gait analysis, and it is a powerful tool for collecting large amounts of outcome data. This is a figure from a paper published by Marks and colleagues a couple of years ago, where they were addressing the question of the myriad variables we can get from gait. In this case, they evaluated 459 variables and submitted them to a principal components analysis, and found that by doing so, there were essentially 3 principal components that explain 54% of what is going on in those 459 variables. And this leads very readily to a question, which is, do we need all the information that we get from instrumented gait analysis? Or is there, perhaps better put, can we distill it into a single number that can be more easily interpreted by a clinician, by the layperson? Could it be could it be a number, just as we have an understanding of where our blood pressure should be? What is a healthy range for our blood pressure? Do we have numbers that would tell us that our gait is within a healthy range, or that we're in a marginal state where there could be problems, could be predictive of problems? Another need is to be using this behavior as a predictor of health conditions, recovery of function. So I do a great deal of my work looking at recovery of function in people who've suffered a stroke. Also to be looking at physical capacity, or gaining an assessment when we observe someone. Yes, they may be getting around on their own, but at what level of their capacity, how much is required for them to be performing at the level that we're observing. And another huge need in our field that I'm pointing to is that we need methods that can allow us to move outside of the dedicated laboratory. So stepwatches and wearables give us some information, although it definitely is a conversation in our field right now, working on the accuracy and the validity of those numbers. But let's just suspend disbelief and agree that we are able to capture movement and activity outside of a dedicated lab. But do we have kinetics? Do we have the ability to get kinetics, the forces? And the question that I'm trying to ask is there some variable or some set of variables that we can capture that would give us a proxy for kinetics? And so I'm going to spend some more time talking about this as we go forward, and why I think that this is such an important element. So what I'm displaying here is a illustration of what we call, for shorthand, A2, ankle power. And looking here on the x-axis, we're going from the initial contact phase of the gait cycle to the next initial contact. And so typically we time normalize our data. And if you think along with me, yourself walking, you initially put your foot on the ground, and then you have a little period of shock absorption where you pull your weight onto the stance limb, a little bit of flexion and extension that takes place. And then as you get towards the end of the stance phase, there is a large power burst. This is power, mechanical power, that is generated at the ankle joint, producing this burst. And this A2, or ankle power, is responsible for forward propulsion in walking. And it is also important for putting kinetic energy into the gait cycle. If there is sufficient ankle power put into the gait cycle at the right time, the swing phase of gait is essentially pendular motion, or essentially passive. And so we see that if this particular parameter is generated and timed properly, it really sets up a lot of important aspects of what we consider the normal walking cycle. So here's some data, and I'd like to focus first on these data in the black. And what we have here is walking speed on this axis and ankle power, what I just showed you, on this axis. And the black dots are a series of healthy controls, people like me, people in my lab, your family, you, any of us who haven't had a major injury. And what this is showing simply is that the faster you walk, the more A2, the more ankle power you produce. And this is what allows me often to say it's just a matter of physics. So A2 scales with walking speed. These additional plots, the orange are from people with stroke. One of the primary impairments, or very observable impairments in people with stroke, is that they walk slowly. They tend to walk asymmetrically, and they definitely tend to walk slowly. And so if we look at them walking at this abbreviation is self-selected walking speed, we find that not only do they walk, the range of speeds that they achieve is lower than in healthy controls, but if we go one-to-one, we find that they are not generating the same amount of A2, ankle power, during self-selected walking. And so this leads to a question of, do they have the capacity to do that? The blue data are the same individuals walking. They're instructed, walk as fast as you possibly can. We want you to walk, not run. Imagine that you're in the middle of a crosswalk and the traffic light has changed. And so we can all picture ourselves in this circumstance. We see that some of these individuals are able to generate a faster walking speed, but even those who are able to achieve, this is a range of what we consider normal walking speeds in healthy individuals. They can, if stroke survivors can achieve that, they are nowhere near generating a normal ankle power. The question is, how do they do that? The short answer is through compensatory mechanisms. But this allows me to make 2 points. One is, if all we do is study walking speed, we really are missing a lot of critical information. And the second is to emphasize this point of how important ankle power is to the normal cyclical function of human walking. Just give a picture of that once again. And in this plot, these are now, this is a different data set, but what we're looking here is self-selected walking speed again versus ankle power. The markers in yellow are stroke survivors, and the markers in blue are healthy controls. And we find that at the higher, so this linear relationship that I showed an image or two ago is more or less present. And there is some interdigitation of controls and stroke. And that occurs, we would call these people high functioning stroke. Some people really have not so much impairment, but they may fatigue more rapidly, or they may have a more subtle gait deficit. But I want you to step back for a minute and come along on this with me, because when I look at these data, I ask myself if there are subgroups. Is there a subgroup here? And if there's a subgroup, I think we could imagine there's a cluster of data here, there's a cluster of data here, another. But then where does this person belong? And where does this person belong? And I'm a big believer that I really need to be informed by my data and not be forcing my data to tell the story I want. And so these types of things trouble me. And then I also look at the fact that we have this large zone of overlap between healthy controls and stroke survivors. And I know, of course, that their gait speeds their gait speeds for certain are not comparable. So one of the first things we did was said, well, instead of just measuring walking speed, we know that there are two spatial mechanisms, that spatiotemporal mechanisms that allow us to adjust walking speed. One of those is step length, and the other is cadence. And step length, of course, scales with body stature. And it's not done as much in the adult literature as in the pediatric or the developmental literature, but there is a relevance to normalizing data by leg length, which is a proxy for body stature. And if we do this, I found that our subgroups became more distinct. And that is somewhat fulfilling. It's a better answer. This would tell me if I'm looking at people with stroke, I might have three different functional levels for stroke recovery. And I might want those insights. There certainly could be neural mechanisms that are differently impaired, or the severity of those impairments could be different. So this was a fulfilling answer, but I do also note that we have this area of rather considerable overlap between some of our healthy controls and some of our stroke survivors. But I do want to note this one person who I was wondering which group belonged to really did belong in the low group. Okay, so this led us to consider, well, there were two things that pushed us into this next step. And so the one piece is we know that we have different functional levels, and we want to be able to assess that. We want to be able to assess it efficiently and also unambiguously. And one of the challenges that I face is I have at my Access an instrumented motion analysis lab where I can get quantitative data and also have the luxury of time to work with it. But if I talk to a clinical audience, they do not have this luxury. So some of the information that I have is not accessible to them, and the conversation really stems on an intellectual level. The other challenge with it is that a lot of times the clinical audiences don't really understand, they haven't had the background to fully understand and appreciate, for example, why I think A2 is so important. And it ends up really changing the conversation of, like, you work off in a rarefied environment, we need you to talk to us in our own language. And so this led to a challenge, sort of challenge issued from the clinical community to say, could you, if you don't believe that gait speed is a good enough measure, could you give us a measure that is accessible to us and is informative and would provide greater sensitivity as you're arguing? And so this led us to this adventure, which in the first phase we had very sophisticated nomenclature for it. We called it the Gait score, and it had 4 components. And the first was, we certainly incorporated their self-selected walking speed, but we did normalize it to their leg length for the reasons that I've shown here, that I think that it really helps to make these distinctions among people's actual function. The second is we asked them to do 2 walking tests, their self-selected walking and then their fastest walking. And so this is observable really in pretty much any environment, but the instructions are take a walk as you usually do, take a walk like you're walking in the park and nobody is looking at you. We're going to time you while you do that. The second task is, as I described ahead of time, imagine that you're in the middle of the crosswalk and the traffic light has changed. So we take the difference between those 2 walking speeds, again, normalized to leg length, and report that difference. There are people who look simply at the range and consider this a modulation score and argue that this could tell us something about their functional reserve. We really generate a lot of information from looking at this fastest comfortable condition. We are essentially able to get people up to the boundary of what their capacity is. And we find individuals who just scale their walking speed with really little effort, and others who have to recruit every compensatory mechanism under the sun in order to get across the street in these per these instructions. All right, our third component is ankle plantar flexion, that ankle plantar flexor power, that A2 that I've told you about. You can think about it as the oomph that we put into the gait cycle. And that oomph is really, I will emphasize, it's not just forward propulsion, but it sets the leg up for the swing phase, for the step length that they're able to take, and for the ability to position the limb. So this particular component ends up being the Achilles heel, our Achilles heel at the moment, because we require the instrumented motion lab. And so this remains one of our challenges that we've not yet resolved. We're seeking a surrogate measure for ankle plantar flexor power. And then our fourth variable is the change in the swing limb step length when they scale their speed between self-selected and fast. So put your hands on the table in front of you. Think of your right hand as being the stance limb. And if you put your left hand in front of you, that's the swing limb. The swing limb is going to go and put its foot on the ground. There's a period of time that the stance limb is in what we call single limb support. It's supporting all of the body weight. There's a balance function that's involved in that. There's a stability function that's involved in that. So when you take a left step, when the left leg is in swing, the entire body weight is being supported on the stance limb. And there should be a difference in that step length when one goes from self-selected to fastest possible walking. And the reason that we picked this variable is for the ability to get at balance and stability. It also tells us about strength in the limb, the ability to support the body weight, tells us about pain. But if you can come back with me for a minute and think about a person with stroke, if this is their paretic limb, their more involved limb, they're going to be weaker. They may not have good sensory integration. They may lack that ability to support their body weight. And so the adjustability of step length between self-selected and fast is going to be limited, because somebody who is very impaired is really not going to be able to tolerate much time on the stance limb. Okay, so we have these four variables: self-selected walking speed, difference between fastest and self-selected, fastest comfortable and self-selected, ankle plantar flexor power, and a change in step length to the swing limb. And each of these intended to give us a component of what we would call gait function. Okay, so I noticed there are a couple of questions, and Corey, are there some that, okay, so there's a question from Dave Jaffe and saying, the question is ankle power measured for their good leg or their affected leg, or an average of both? We measure it in both. When we have them in the instrumented lab, we are able to get both limbs, but the more relevant to the conversation, where I've been showing you this impairment in stroke, is the paretic limb. And so the diminished power is in the paretic limb. A question from Holly Jemison asking, would it make sense for the sixth vital sign to be more general than gait speed, perhaps motor speed? I'm going to, I think this is worth much more discussion, and I think I would like to leave that to the end if we could do that. Okay. So this was a diagram to provide an illustration of the what's happening in the stance limb when we have the change in step length. And just up here in the top left corner now, these are the four components of our gait score. Plotting this here against A2. This is the A2 ankle power that the individual is generating And to take Dave Jaffe's point, if they were somebody who had had a stroke, it is their paretic limb A2. And if they're a healthy person, we flip a coin and give them an experimentally, we just target one limb and report the number in one leg. So again, yellow here are stroke survivors and blue are healthy controls. And I'll remind you, we had clusters when we were looking just at gait speed, but we have these by going to this 4-component score. It seems as if we have made greater distinction in the functional levels of these individuals and spread them out over the range of ankle power values. One of the things I failed to mention here is that we have these 4 components. We score each of them. We give each of them a score from 0 to 3. The 4 components total in our Gait score to a total of 12 points. And so that's what we have on this axis, assuming if we're referencing normal function, that a healthy individual like you or me would get 12 points. And there would be some reason if individuals were not getting the full marks on all 4 components of the Gait score. So what we're showing here is, in fact, our premise is premise is more or less confirmed. I think one of the important things is that in terms of our subgroups, we were able to create this dispersion. We do have stroke survivors who are very high functioning and function and look almost like controls. And then here in this cluster that I've numbered as cluster 3 is where we had a surprise. And the surprise is the number of healthy controls, ostensibly healthy controls, who did not get full marks on the Gait score. And so this was one of those moments in science where you say, well, maybe my idea wasn't so good. Oh, well, maybe we have a data entry problem, or maybe we calculated something incorrectly. And long story short is when we went back and really looked carefully at these individuals, we found that each of the 5 individuals that fall in that cluster had an underlying condition. They had come to the lab to be controls for a study of individuals with stroke. They met our screening criteria. There was no reason we shouldn't have studied them, but we found that they had some underlying condition that they hadn't told us about when we took their health history screening. Furthermore, our team, who are all quite expert at observing gait impairment, were unable to detect any gait deviations, and they all walked at a normal walking speed. So this is where it got interesting. And it got interesting because these individuals had different pathologies, emerging pathologies that ranged from a musculoskeletal condition to a degenerative respiratory condition that was diagnosed in that individual perhaps 6 weeks after we had studied him in the lab. And so we were able to go back and say, well, which component of the 4 components on a Gait score, which component is responsible for them to lose points, and it does differ by their pathologies. Now, an additional piece, I've sort of teased you with the idea that we like to study the nervous system. So these are motor evoked potentials generated by TMS. These are targeting the plantar flexor muscles. And these individuals were doing either an isometric, a static plantar flexion, or a dynamic plantar flexion where the joint was allowed to move. And we expect that the volitional drive is much greater in the dynamic condition. And so we expect to see something like this or like this. The dark blue trace is one where they were performing dynamically and they were able to modulate the size of their motor evoked potential. So we see individuals here who what you see is what you get, and all the way up the continuum. I'm going to take this back and make the point that these clusters, these 5 clusters that were captured by the Gait score, map with integrity of their corticospinal tract. And so, we're onto something. We have a brain behavior correlate and reasons to understand the impairment in their plantar flexor performance, but also showing that it has a functional relevance in their gait. So I'll be honest with you that I was really doing this experiment to get the TMS data. And when I went and talked to the clinical world, they were a whole lot more interested in the Gait score. And part of the interest in the Gait score was this bit of walking speed really doesn't tell us enough. And you seem to have identified some important variables here. So, and it does a good job. The three of these, the walking speeds, the change in step length, can all be measured relatively simply. I don't think they can really be measured with something like a wearable, like an Apple Watch or Fitbit, but they can be measured with a device called a gait mat, something that can give you spatiotemporal parameters, but it is still an instrument that's required of that. The plantar flexor power, we require the full instrumented lab. So this was all well and good. And I want to make the point, and I think any of you have had enough time to look at this figure and realize that we have about 30 or 40 people in here. It's not a huge sample. In the world of biomechanics, this is a credible sample. This is a lot of work for us to get to this amount of data. But in terms of epidemiology or larger statistical types of samples, this is not such a large sample, and we absolutely acknowledge that. And so I give full marks to a PhD candidate in my lab, Carolyn Banks, pictured here, who has always been one to push us on our mathematical methods and our statistical methods. And she came to me and she goes, you've been doing this for a while. You have instrumented gait data that are available from a number of studies that you've done over the years. Could we concatenate these data sets and then have a larger sample? And so she did actually a great deal of work, went through studies that we had collected over 10 or more years. Some, not always using the same motion capture system and things like this. So as I said, a fair bit of data cleaning. But the idea was we had developed this Gait score in this small sample. Could we test it and validate it in an independent sample and see with larger numbers if it still held? And so this is in fact what she did. She was able, by the time we got to the end of the day, to identify 87 individuals with stroke and 36 healthy community dwelling individuals. She used the 4 same components, and then one of the steps of validation was to validate against typical clinical measures that are used for gait and mobility. So the Fugl-Meyer is a measure of motor impairment, and that's performed. It's specific to post-stroke hemiplegia. The Berg Balance Scale is a very robust measure of balance, and it was developed for older adults and then later validated in stroke. The Dynamic Gait Index is intended to get at transitions, head movements, changes in posture, but has been validated also both in older adults and in people with stroke. And then the Short Physical Performance Battery is exactly what the name implies. Short and it's efficient. It was developed by Jack Garalnik decades ago, and it has been used enough now that it is a very accurate, can be used as an accurate predictor of all-cause morbidity and mortality in older adults. So we thought this was a good set, and basically she correlated correlated the score against these 4 measures, found that the correlations were strong, so we had face validity, and found as well that the correlations were not driven by age, and they were not driven by stroke chronicity. And so it does tell us in the aggregate that we're coming away with a construct that is telling us about their mobility or their motor impairment. So in this sample of about 130, she further did a factor analysis and found that the data loaded on two factors. So one factor we call speed and power, and that was comprised of ankle power and self-selected walking speed. And the other was, we named it as adaptability. It was comprised of the speed modulation and the change in swing limb step length. And the correlations were strong. And even though, again, it's relatively small sample for doing a factor analysis, the statistician took a look at it and said, meets the sniff test. And I really think that you do have these two factors within this construct. And these are two of the critical functions of human walking. So we're now at a point, we've developed a measurement assessment. We have validated it in an independent sample. Part of what is exciting about it, I think to me, one of the most exciting things about it was the ability to detect these individuals who were ostensibly healthy, but when we really dug deeper into their background, we found that they had a subclinical level of pathology. And this, I think, is the type of value of this type of measure, is that we're looking at something that is non-invasive. It can be, in theory, performed in a clinical setting, in a community-based setting. And it is the type of thing that with sufficient data to validate it in larger samples, we could start to provide it as a clinical tool that might be predictive, a predictor of oncoming pathology. And so this led to a scheme that we have referred to by shorthand in the lab as mobile gait assessment. And over some conversations of how would we go about doing this, and keeping in mind we come from a tradition of studying 30 people is a lot. How would we go about getting larger samples in order to get the data efficiently and have the generalizability and the diversity that we need in our sample to have to really stand on this as a measure? So it led to the construct of mobile gait assessment, and the idea of it being, could we set up something that would look essentially like a TSA screening station, where a person would come in and be greeted, we would consent them, have them fill out a health questionnaire, we take some measurements, maybe take some of these clinical tests I just discussed, and then using different forms of markerless or inertial motion sensor type of measurements, could we measure them? Also, one of the tools we have in our lab is a pressure sensitive walkway called the step scan, and I'll show you this in a moment. And so they would come, they would navigate through all of these different stations. We would get motion data, we would get spatiotemporal data, we would get pressure data, and these would then provide us with information for our 4-component score. And we had some fun thinking about, would we go to a health fair? Would we go to the atrium of the major hospital, local hospital? Where would we go someplace where we could find people who would just be willing to walk through this, because it would take them 5 or 10 minutes, and we would garnish a lot of data that we could then work from. Okay, so keeping in mind on this mobile scheme, there are walkways that people can walk over, but they do not provide us the force, the actual force measurement from a force plate that can give us A2. And A2, A2 is sensitive and something that we like. So as I said, this is the Achilles heel of our measure at the moment. This is, here I'm illustrating our pressure sensitive walkway. And so, in some ways just looks like floor tiles that you put together, but it has a very high resolution, high density, high resolution sensors in it. And these are footprints of somebody from the lab walking through here. And so we can get a distribution of how they are loading, how they're loading the foot as they walk. We get all the spatiotemporals, the step length, the cadence, all of that, but we're also getting this loading pattern. And wait, there's more. We get this, and this is in pressure, units of pressure, but this green trace right here looks very, very similar to what we get off of a force plate that is installed in a fixed instrumented lab. So more detail than you really care to think about, but the takeaway is this looks very, very similar to the vertical component of what we can get from a force plate. So perhaps there's some useful information there. So a project we did with a medical student the first year we were here, they said, well, can we get something just from this force trace? And that's what I'm illustrating here. And I look at, this is the language of Gait for people like us, but what we've done here is annotated this. So this part of the curve is when you're loading the limb. This part is when you have both feet on the ground. And this part is that late phase of stance when you're pushing off and you're in single limb again. So we took the slope of this, we measured the slope of this to see if that could give us a surrogate, or help us to find our way to a surrogate for this A2 measure. And the data that we had, this is a sample of about 30 people. This is everybody, of course, lots of variability. This group of people all self-disclosed a prior musculoskeletal injury in the leg. And interestingly, the range of their walking speeds was lower, and the slope of this late phase of force production was lower. And so very preliminary data, but this was one of our first rollouts of the mobile gait scheme. And so we thought, well, perhaps we're on early days, but encouraging science. So our next move was about a year ago. We acquired, well, maybe it hasn't been quite a year, several months ago, we acquired technology to do markerless motion capture. And so this is a picture of one of the first times we set this up. We're basically using commercial grade cameras, set them up in a large volume. We still need to use a calibration object because we have to synchronize the cameras. But the advantage to this is you do not have to put markers on people during kind of COVID. We don't have to touch them, but we just have them wear their own clothes, their own shoes, and use we have a software that is called Thea that uses an AI algorithm and is able to synchronize the information from the cameras, build a 3D model, and that is the source of the skeleton that is overlaid on the person walking here. So just do this again to show another view, same person, same day, another view of it. We can do this up to 8 times. And what this leads to very quickly is we can get joint angles. And so these are joint angles at the hip, the knee, and the ankle. Each of these lines is a step cycle, right and left leg. But you immediately can see that we have a lot of information here about this person's walking pattern, and that can complement their walking speed. But we can now tell about, are they getting full normal range of motion? Are they getting how variable or how stable is their walking pattern? And what are the differences with them across between and within conditions? It's just a joke that I like to make, but armed with markerless, we get out of the lab and you never know when to expect the Spanish Inquisition. And so we have been setting up in a number of different places. This is the lobby of the med school, Sacramento. This is the pavilion on the Davis campus. Just will do this again. This is a video of the same person that you saw a couple slides ago, different day, different place, different clothes, different shoes. And we're able to still get data. And here are these curves that you are now familiar with. And this overlay is showing that these curves are reproducible, even if we do them in multiple locations, or if people are not wearing exactly the same attire. This is a real game changer for the world of motion analysis, because traditionally, not only do people come to the lab, but we spend an hour and a half instrumenting people before we can study them, and lots and lots of high maintenance work on that. Okay, so we have spent a lot of time convincing ourselves that we can get good data, and this has now enabled us to move forward to a project that we call WILD, or Walking in the Living Domain. And I think you can connect the dots with us there. We're out of the lab and we are now able to collect data with people. We can get closer to people and make it more feasible for them to participate in our studies. So this is Alumni Hall over on the UC Davis campus. We set up here for a couple of weekends and had people come through. This is the lobby of the Midtown Clinic in downtown Sacramento, or Midtown Sacramento. It's put us very close to a clinical population. And in this illustration, we're showing both their self-selected and fast walking, but the gray shaded area back here is we're referencing an individual subject's data to normal. And these are normal subjects, our database of normal subjects that we have studied over time. So this gives us a chance to interpret our people deviating from normal and where and how, and providing us this finer grain level of information. So I just want to wrap up with, this is a project that we're working on right now. The part of the goal of WILD is to get at this point of, can we get gait analysis distilled into a single number that can be easily interpreted? So we're continuing to build the ABLE, but using now our portable technologies as a way to get us outside the lab and closer to people so that we can get larger samples. We continue to be interested in looking at it as a predictor, either of emerging health conditions or other more traditional interpretations of clinical measures. And we continue to work on developing methods to be outside of the dedicated lab. And one of the challenges, as I've made several times now, is how do we get kinetics and how do we get a proxy measure for that? But we have started to partner with people who do machine learning to see if there are ways that we can inform, use machine learning to inform us on this question. I'm going to stop there, not without thanking my team, who are the people who do the work. And thank you for your time and your listening. And I'm going to shift over here to questions. Oh, Eric Sabelman. Hello, Eric Sabelman. Eric Sabelman answers no. Nobody's tried it lately. We're working on it. Nisha. The neural control that underlies aspects of the stroke. I'd be happy to discuss that. I don't think I have time today. That's really my day job. Okay, there's that.
Great. I think we have another couple questions.
Okay.
So I have one question. How sensitive is the methodology to detect between normal gait changes and changes that might lead to, say, falls? Have you done much work with falls, and since that's such a huge risk for older adults?
Yeah, so I really wanted just to sort of talk about the trajectory of the project today, and I didn't spend a lot of time talking about falls and risk with falls. But yet the short answer, Corey, is yes, the technology is sensitive enough, and it's all a matter of what are the measures that you take. And so we could be looking at variability in any of these components. Variability tends to be something that's really quite sensitive to fall risk.
Great. Another question, I think this will probably be one of our last ones, is have you worked with clinicians on interventions that can improve strength and mobility that are more on the low-tech side and can be implemented in the home or the residential settings?
That's kind of a sticky wicket question. I, of a time, have done interventions that are higher and lower tech, and I have done interventions where we have been using Gait, instrumented Gait, as an outcome measure. We do show differences. This particular application of using the ABLE and using the markerless technology, no, we haven't gotten there yet. This is work that we're working on right now.
Great. Well, thank you very much for your talk today. It was very fascinating, and just want to thank you for being here.
All right.
And then want to thank everyone for tuning in today, and hope you'll join us next Wednesday with Ahmed Shabir Arif, who's an assistant professor in computer science and engineering at UC Merced. And his talk is going to be entitled, "What if computers could read our lips? Silent speech as an active mode of interaction with computer systems." There'll be a link for more information. And then just to note that the webinar recordings will be on our playlist on YouTube. And so you can go check out this talk tomorrow afternoon. Thanks again, Dr. Patton, and appreciate you being here today.
Thank you for the invitation. Thank you all for listening.